The goal of this procedure is to determine the activity of cytochrome P450s, or CYPs, in cultured cells. CYPs are key enzymes involved in the metabolism of medicines and toxicants, and their expression is not often retained in vitro.
Normal expression of CYP enzymes is often lost when cells are cultured in vitro. Therefore, many in vitro models are potentially limiting relevance to understanding the metabolism of medicines and toxicants. The main advantage of this method is its simplicity when assessing CYP activity in cultured cells. Using probe substrates and inhibitors, different quantification platforms are presented, including mass spectrometry.
To begin, obtain cryogenic vials with cells as described in the text protocol. The protocol provided uses a hepatic cell line as an example. Whilst working under sterile conditions, carefully open the cryogenic vial to release any excess pressure. Then, pipette the contents of the cryogenic vial into a 10 ml tube filled with 9 ml of pre-warmed Williams E medium at 37 degrees Celsius. Centrifuge the solution for 10 minutes at 400 times G at room temperature. After discarding the supernatant, resuspend the cells in 5 ml of proprietary thaw medium pre-warmed to 37 degrees Celsius and supplemented with glutamine. After counting the viable cells as described in the text protocol, seed the cells in a 24-well collagen-coated plate at a density of 60,000 viable cells per well in a final volume of 0.5 ml of thaw medium. After a few days in culture, the cells form a subconfluent trabecular structure.